Good afternoon, everyone. Thanks to the organizer for giving me the opportunity to share some of our recent work with you all. I'm Federico Gaiti, a scientist at Princess Margaret Cancer Center and an assistant professor at the University of Toronto. And today I'll tell you a story at the intersection between cancer biology and neuroscience. We know that tumor progression is fundamentally a process of somatic evolution because genetic alteration generate diversity, and then ecological interaction like competition between clones drive natural selection across that diversity. And then over the years, the field has indeed proposed multiple models of cancer evolution shaped by these genetic changes. But while most cancer are initiated by genetic alteration, the adaptive capacity of cancer isn't fully explained by genetic alone. Tumors are heterogeneous, evolving ecosystem encoded across multiple layers of information: genetic, non-genetic, transcriptional, spatial, and microenvironmental. And so we think that to truly understand how cancer adapt, we need evolutionary model that integrate both genetic and non-genetic variation. So our mission is to map how tumors evolve by identifying malignant cell state, how they interact with their surrounding, and the full range of diversity within a tumor, such that we can then understand how cancer change and adapt over time. One of our preferred model, disease model, is brain cancer, particularly glioblastoma or GBM. GBM is an aggressive central nervous system malignancy with a devastating prognosis. The current standard of care, which consists of maximal surgical resection followed by radiotherapy and chemotherapy, is not meaningfully changed in nearly 2 decades. And so despite aggressive treatment, though, the 5-year survival rate still remains under 5%. So what makes GBM so hard to treat? Well, a major obstacle is for sure this intra-tumoral diversity, because this gives the tumor basically many recipes to try, and increasing the odds that some cell state or malignant clone actually can survive therapy. Even when the initial responses are strong, indeed the tumor often adapt within months to a few years and often recur in a more resistant form. Single studies have made this especially clear because we now know that GBM's tumor are this admixture of malignant cell state that actually resemble very much neurodevelopmental programs, varying along axes such as stemness, metabolism, and lineage like specialization, but often in a way that is partly independent of genetic clonal diversity. There is also another layer to the problem. Surgery these days is guided by MRI visible tumor boundaries, but many malignant cells actually extend well beyond that visible region under MRI. They can migrate into adhesion, similarly in a normal brain tissue. And then radiotherapy and chemotherapy often fail to eliminate this more infiltrative cell, which then ultimately we think are the one that seed actually the recurrent tumor. And so this suggests to us that infiltration isn't just a fixed genetic trait. It may be a state induced actually or reinforced by the surrounding brain microenvironment. And so in our work, we ask a simple question: how does the tumor microenvironment remodel the epigenetic landscape of GBM malignant state to promote now an infiltrative phenotype? To answer this, we partnered with Dr. Gelarezade at the time at the University of Nectar and sample anatomically distinct region, the peritumoral brain, tumor edge, and tumor core, guided by intraoperative neuronavigation and preoperative MRI images. We then profile this region with single-cell nucleus multiomics and spatial transcriptomics to better characterize the phenotype of the infiltrating malignant cell state. We recovered the expected major cell type, including malignant cell and then diverse microenvironmental population. And we also detect, most notably, malignant cell that already infiltrate the peritumoral brain beyond, again, the visible MRI board. And we confirm that what we call malignant cell, indeed, that infiltrate the normal brain tissue, are indeed malignant by reconstructed copy number aberration profile, leveraging the single cell RNA sequencing data. And here we can see that by doing this exercise, we can recapitulate the very classical GBM hallmarks, which are chromosomal 7 gain and chromosome 10 loss, which contain P10. And so with this, we can now confidently label cells that are non-malignant and cells that indeed possess these classical hallmarks, and we label them as malignant cells. Importantly, then, what we saw is that malignant states were not evenly distributed, because progenitor-like oligodendrocyte progenitor cell, we saw they were enriched more toward the periphery of the tumor, while the tumor core was more populated by malignant cells in these more mesenchymal-like programs. We confirm again the presence of this infiltrating oligodendrocyte progenitor malignant cell in normal brain using then multiplex immunohistochemistry. And here we can see that SOX positive, SOC2 positive cell in the stroma actually co-express very classic malignant markers such as OLIG2, ASCL1 and DLL3, which is a notch ligand. And here again we show that these malignant cells are more preferentially, infiltrative cells are more preferentially visible in the infiltrative area of the tumor. We then compare peritumoral malignant cell now to their core counterpart to learn what could be unique feature to this infiltrative cell. That standout signal was this neuronal activity and synaptic associated transcriptional program that is selectively elevating in infiltrating cell. And this is again very much consistent with the emerging cancer neuroscience model, where it was shown that tumor cell can hijack neuronal signaling to now support invasion. To confirm the communication now between this peritumoral infiltrative cell and neurons, we use also in situ spatial transcriptomic and again multiplex immunohistochemistry to again show that this infiltrating OPC-like cell colocalize more frequently with neurons. We can, by probing a little bit more the spatial transcriptomic data, we can indeed see that the infiltrating GBM cell more frequently actually colocalize with neuron, especially in the peritumoral cortex, and they are very actually distinct from the type of interaction that we can capture instead in the tumor core area, which are prevalently driven by more malignant state in this mesenchymal or more like hypoxic phenotype. And so from this cell-cell communication prediction, we could also confirm the glutamatergic nature of this neuron-tumor interaction, and also found a neurexin signaling axis, which again, this suggested to us that this tumor cell may actually form proper functional synapses and integrate into neuronal circuit to increase their migratory and invasion capacity. And again, this is very much consistent with seminal finding in the field of cancer neuroscience, but from animal work, and here we are showing it directly in patient in human patient sample. In vitro, then we also demonstrated that human iPSC derived neuron co-culture now with patient derived glioma stem cell line can also indeed enhance the migratory capacity of this glioma stem cell. And so this for us was again the functional validation of the observation that we gather from the omics data. So we know now that neurotumor crosstalk may now induce again some reprogramming in this invasive high and malignant cell. We then speculate that this malignant cell may now exhibit actually distinct epigenetic landscape that would now reinforce this invasive and infiltrative phenotype. And so we ask a simple question as a next step, which was which factor regulates the infiltrating OPC malignant state? So to answer this and trying to uncover the key epigenetic regulator of this phenotype, we now leverage match single nucleus transcriptomic and chromatin accessibility data. And here we identify transcription factor including TCF12, TCF3, TCF4 and Z1 as potential regulator of this invasive high OPC-like cell. Functionally, we also show that Z1 disruption by CRISPR knockout significantly now reduce migration in a transwell invasion assay, and we also see this reduction in the invasive infiltrative phenotype by qPCRs, which again support more a causal role this for this transcription factor. And an observation we made is that this same transcription factor, again the TCF family, Z1 and ASCL1, are also very much a key transcription factor in normal oligodendrocyte differentiation. And so this at the time suggests to us that perhaps infiltrating GBM cell may actually hijack normal developmental programs at their own advantage, and in this case would be to preferentially drive an infiltrative phenotype. And so what we did here to probe this hypothesis was to reanalyze a human fetal brain chromatin atlas to show that actually a transcription factor driving the more uncommitted OPC-like state in this invasive malignant cell very much mirror those that regulate early oligodendrocyte development in normal physiology. Indeed, we know that in normal brain development, oligodendrocyte progenitor cells progressively lose their capacity for migration and axoglial formation as they mature. And so the model we have in mind here is that infiltrating cell actually hijack OPC lineage transcription factor to maintain themselves into this more uncommitted state, enabling them to hijack this normal developmental function for brain invasion and have higher migratory and synaptic capacity. On the epigenetic side, then further generation of single cell methylation dataset suggests that this state is actually locked in, meaning that a variety of key oligodendrocyte differentiation gene were hypermethylation, meaning that they may limit now maturation and stabilize this invasive uncommitted OPC-like program, and then almost suggesting to us that in this infiltrating OPC like cell are maintained into this more immature undifferentiated state. We then showed that by knocking down ZEB1, we can actually now shift the phenotype of this malignant cell to a more differentiated and presumably less invasive phenotype. So for us, this was again the first live proof of concept that we now have the possibility to intervene and intercept and change in a way that the trajectory of these malignant cellular state phenotypes. So to summarize, we showed that glioblastoma plasticity is driven in part by epigenetic mechanism that maintain this infiltrative, invasive oligodendrocyte progenitor-like malignant cell in this more uncommitted immature state, and because this is the state that would enhance their migratory and synaptic properties to adapt to the peritumoral microenvironment. This work was published last year, so feel free to take a deeper look, and we'll also happy to discuss further during the Q&A section. And I'll just like to conclude by telling you why do we think this work is important. Well, clinically, obviously, this now point to us to the idea that we can think about combination strategies to try and limit and delay recurrence by pushing the cell toward a more differentiated phenotype that would reduce invasion, but also we are thinking about how to manipulate and disrupt neuron tumor crosstalk, and specifically targeting region of high functional connectivity. And so we have now these two follow-up direction where we are trying to target more the interaction with neuron, but also target more the molecular phenotype of this infiltrative GBM cells, specifically at this minimal residual disease stage, which we think is the actual window of opportunity, and we have the hope that this would delay and at some point prevent a recurrence. So with this, I like to conclude. Thank again the organizer for giving me the opportunity to share some of our recent work with you, patient and their family that made this study possible, people in the lab, our collaborator, all of the funding agency, and you for listening, and happy to take any questions.